these sensory receptors, and now we're talking about sensory receptors in terms of neurons. Because again, receptors, that term used in the book, that often they kind of flip between receptors by talking about an entire neuron or a part of dendrite or talking about a protein. So I'm going to talk about cells for this slide. So there are all these different stimuli that you can detect from your environment and your own body. And there are specialized cells that detect each of these stimuli. So things like painful stimuli. Actually, this is a really useful prefix. So whenever you see this term nociceptor, I believe it comes from the same root word as noxious. So when something's noxious, it's kind of like offensive and toxic, right? So if you have a really noxious odor, it's like, oh man, that really smells really horrible. So think of the way I remember it, like nociceptors. If you smell something that's so noxious, it causes you pain. This is nociception. So again, that's the way I remember it. But things like thermoreceptors, like thermal, refers to temperature. So all of these are special types of neurons that detect each of these stimuli. And there is some overlap, like things like nociceptors. You can have nociceptors that are also chemoreceptors, but thing is that some of these you don't want to overlap. Like, do you want, temperature stimuli activating baroreceptors? So if that happened, well, maybe when you step outside into a cold room, it feels like someone slugged you or something. Or would you want it where if you just have a change, take a dip in, or change your oxygen and carbon dioxide, that causes you to feel like someone's like rubbing against you? Well, that would be kind of weird, right? So things that you don't want these to get mixed up as well. That's why they're all specialized. So I want you to definitely associate these stimuli with all of this vocabulary here. That's definitely what you should know. So again, stimuli activate these receptor cells. That's why your book doesn't list it this way, but I'm trying to really show you that we're talking about cells in this table here. Now, the thing is that, again, your book tends to switch between sensory receptors and referring to a cell, and also protein receptors. So these are small proteins on these cells that actually detect physical phenomenon. So the thing is that sensory receptor neurons, they have protein receptors, and often these protein receptors are some variant on an ion channel. So things like thermoreceptors, well, they might have temperature-gated ion channels that open in response to some change in temperature, whether it's getting hotter or colder. And chemoreceptors, they can also have ligand-gated ion channels. So remember that how acetylcholine opens ion channels. That's an example of one of those protein receptors. And again, this is on a receptor neuron, or sometimes a receptor cell. And G protein coupled receptors, I believe that unless they change things, you're probably going to cover that in like sophomore biology or cell molecular biology. So I'll leave that for that class because it is a pretty complex pathway. And then mechanoreceptors, these are mechanically gated ion channels. So again, some ion channels, they're shut, but when you change the membrane, it kind of stretches the membrane and opens these channels because again, these channels are embedded in the membrane. So actually, let's go with that example. So here we have a mechanically gated ion channel, and here it is in the closed state. And remember, in a resting neuron, you typically you have a higher concentration of sodium ions on the outside than you have in the inside cytosol of the cell. Well, say something, so right now it's closed and something touches it. So your sense of touch often involves these mechanically gated ion channels. If something touches your cells, well, it's going to stretch the plasma membrane or distort it. It's going to also stretch these channels and distort them and cause them to be more open. So what happened here? Well, remember that term membrane potential, and roughly in layman's terms, it's kind of like the electric charge inside of a cell. So what happened here? Well, remember these sodium ions, they are positively charged. We just had more sodium ions flow into the cell. What happens to the overall charge inside the cell compared to the outside? Well, it just got a little more positive. And if the cell was resting and now it reaches this magical number we call the threshold, what's going to happen? Well, now the inside got a little more positive, and now it's going to open these voltage gated ion channels. And remember that special term action potential. So what sets off the first part of an action potential? Well, these sodium voltage gated sodium ion channels opening. And now you have that action potential. Now you have that signal traveling along the axon of this receptor neuron. So this is what happens. So this is just an example of how, again, your sense of touch is activated. Again, some sort of mechanically gated ion channel, this protein on a neuron is opening in response to stimuli. Then this sets off an action potential. So it all now comes together. This is where the part in the semester where everything starts to link up with each other. And nociceptor ion channels. So remember, nociception refers to the sense of pain. Now, is there just a pain protein? Is there just one type of pain stimuli? And many things can cause pain, right? So the interesting thing about nociceptor neurons is that they can use a variety of ion channels. So things like temperature-gated ion channels. So if you touch a hot stove, that's painful, right? Or if you stick your hand in, or if you have like a hot day and you have the ice bucket and you have all the cooler with all the drinks in it, what if you keep your fingers in there too long? Well, it gets a little painful. It eventually gets numb, but it's kind of painful if you go to a very cold place and you touch a cold pole. That's kind of painful as well, right? So temperature-gated ion channels can be on nociceptor neurons. So again, some change in temperature causes these to open. All these sodium ions flow in, and this causes it to reach that threshold potential. Or it can be mechanically gated. So if something pokes you, like something like a needle or sharp object, or slices you or punches you, I mean, this is all going to cause some sort of distortion in a cell membrane. These are going to activate mechanically gated ion channels. So these are going to, again, set off the change in membrane potential that causes an action potential. And things like ligand-gated ion channels, like things like pepper spray or eating too much of that capsaicin. Or have you ever had that Korean ramen where it has this angry looking chicken holding a bomb? And I couldn't even eat one bite. I was in such pain. It felt like 10,000 needles were in my tongue. And all I could taste was pain. But what's happening is that it's binding to this ligand-gated ion channel, triggering all these nociceptor neurons on my tongue and sending it to my brain. So, I mean, yeah, look, I just like gave it to my, yeah, I just gave it all my packets to a friend who actually likes that stuff. I can't handle. But basically, what's happening, chemical is binding to these ion channels, opening it, and causing the changes in membrane potential that cause action potential signals.